We're talking about cell division. Yep. Oh, yeah. All right, so we're talking about how cells divide, and we're going to first talk about how bacteria divide, which is a little more simple of a process because of the structures of bacteria. So what are the characteristics of chromosomes in bacteria?
Circular.
Okay, it's circular. Good. All right, so this makes their replication easier. So what they have in bacteria is they have a site called the site of origin. Okay, and these steps in binary fission are a little bit different than what was in the video lecture, just because when I went back and looked at it, didn't make total sense. Okay, so first step is replication begins at the origin of replication, or the site of origin, something of that nature, origin site. Okay. And this is a specific sequence which is recognized by enzymes of the bacteria, and it'll start unwinding the DNA and making copies of it. Okay. And so, because it's circular, there's no beginning and end to the chromosome, and so replication can continue in both directions. Okay, so it'll start adding new pieces of DNA in both directions. Until it reaches all the way around and finds another specific sequence which is designed to terminate replication. And what it looks like once it gets around there is something like this. And it has that termination site right there in the middle. And then there will be two origins because we made a copy of the origin as well. Okay? Okay, so the last little section to complete is the termination site, and then you have two separate chromosomes. Those chromosomes then separate to opposite ends of the cell. Okay, and the cell also elongates. And then finally, the process of septation, where a septum forms. And then that pinches off, and then you have the formation of two complete cells. Each with their own circular chromosome and are exact replicas of our first cell that we had. Okay, so because prokaryotes, bacteria are They just have that one chromosome. They're fairly simple. This can occur at a pretty rapid rate. You get 1 into 2, and then those can turn around, and as long as you have all the nutrients required, you can make 2 into 4, and so on and so forth. All right, any questions about this? Yes?
Does it ever happen where the cell tries to split with the chromosomes on each end, but something went wrong, and so it has both chromosomes on one end or something?
Yeah, I would imagine that happens. I don't know specifically, but the one without a chromosome couldn't do anything. And the one with 2 chromosomes would have a redundancy. No, because these are bacteria, so. But they are twins. They are essentially identical, but they are not humans. Okay, so I have an example of this little video of binary fission we'll watch here, and it gives you a little time lapse here. All right, so you can see it's starting with 4 or 5 bacteria, and each one is elongating. During that elongation process is also where the chromosome is replicating. We can't really see it though, but you can see septation occurring. Okay, so when they're done, then it pinches off, the septum forms, and it breaks off into 2 cells. Now what's kind of cool is this one right here, it's getting really long, and you'll see actually septation occurs in multiple spots. So that means it actually replicated the bacteria multiple times before it separated off. Yeah. So as long as you have a continual amount of medium or nutrients for these bacteria, they'll just continually divide one after another. And that's how they basically have unrestricted growth, unless there's something else eating them or something else in there killing them. And this is how an infection works as well. Okay, more fun stuff like that if you take microbiology. Okay, so we're just going to do a little bit of background of terms that refer to chromosomes, and then we'll talk about the cell cycle, which is going to set us up for the stages of mitosis, which is what we're going to do on Friday. Okay? Okay, so I drew an amorphous stick figure here. But to represent a eukaryotic organism, okay? Eukaryotes have 2 copies of every chromosome in their cell. And that is, here I'm going to write on this side. Diploid, okay? And represented by 2N. Okay, so that means you have 2 copies of each chromosome. Okay, why do we have 2 copies? Yeah, so you get 1 from the sperm and 1 from the egg. Okay? So the diploid cells in the body are also called somatic cells. Okay? So these are the cells which carry out the functions of the organism. So your hand, your foot, your hair, all those are somatic cells. But there are specialized cells which are haploid, meaning they, or 1N, meaning they only have 1 copy of each chromosome. Okay, so the process by which you get 2 copies down to 1 copy is called mitosis. And this occurs in gametes, or also called sex cells. Which are the sperm and the egg. And then the process by which you then have a diploid organism is fertilization, when the sperm and the egg come together, and now you have a donation of one set of chromosomes from the father and one from the mother, making a diploid organism. What's that? Yeah. Yeah, and then it goes through mitosis. All right, so that's simple enough. You guys all understand that, right? All right, so what's the typical shape of a chromosome? What do you think of when if I said draw a chromosome, what would you draw? A line? Okay. You wouldn't draw an X like this?
Yeah, it's kind of like that. That's exactly what I thought.
Okay, so generally that's how they're drawn, is just little X's, okay? And that's one type of chromosome, but this also is another type of chromosome. And a chromosome that has one of each of its genes copied in there is called a chromatid. Okay, so each section that has one of those, one linear chromosome, is called a chromatid. All right, this over here is also one chromosome, but it has two chromatids. And how it got two chromatids is through the process of replication. Okay, and when the two chromatids are the same, you would call these sister chromatids. And they're the same when they're on the same chromosome. Okay? And this is also called a tetrad. Okay, tetra means four. It has four arms, essentially, which make the X. All right, so there are a few more structures on this tetrad. So what's tying the sister chromatids together? Are proteins called cohesion proteins. Okay, where did we hear the term cohesion before? Water, right? So water sticks to itself. You have two chromatids sticking to each other through these cohesion proteins. And then they also have sites on the outside called kinetochores. Okay, so this has replicated so that it can split. Okay, you doubled the copies so that you could split the copies and then move them and make a new cell. So the kinetochores are the site of attachment for the microtubules, which will pull them apart. All right, so one way to represent the chromosomes of an organism is called a karyotype. And I'm just going to make up a karyotype. In the video lecture, I showed you the karyotype of humans, which has 46 chromosomes in 23 pairs. All right, so I'm going to draw a karyotype that has one chromosome here. This is chromosome 1. Another chromosome here. This is chromosome 2. And this is chromosome 3. So this organism only has 3 chromosomes. Here is the visual array of their chromosomes. And these two are, the red came from the mother and the blue came from the father. And so because these are the same chromosome, just from different parents, these are called homologous chromosomes, or homologs. So these are also homologous chromosomes, and so are these. Okay, and you could also draw those homologous chromosomes as tetrads. Okay, so if I wanted to draw this first one as a tetrad, those would also be homologous chromosomes. They just have 2 chromatids instead of 1. Okay, any questions about chromosomes?
So they can't be homologous if one is 2 chromatids and one is 1 chromatid.
They can. They're still homologous chromosomes. But if you're looking at this one and this one, those are non-homologous chromosomes. Those are different. They code for different genes. Okay? Whereas if your dad was, let's say the gene for height is on chromosome 1, your dad was tall and your mom was short, those genes are going to be the same, in the same spot on those different chromosomes, and they may code for different things, but they're still on the same gene. Okay? All right, the thing we're going to do is the cell cycle. Okay, so most cells, okay, we're going to talk about mitosis, which is the part of the cell cycle where a cell is splitting into 2. And a lot of people think, well, when you talk about the cell cycle, you're talking about mitosis. But mitosis is actually just a small part of the cell cycle. And the end of mitosis is the splitting of the cell, which is called cytokinesis. Cyto means cell, and kinesis means movement. Okay, but most of the cell is in, or most of the time of the cell is spent in interphase. Okay, so that's the rest of the cell. An interphase is split into 3 parts as well. So the first one is called G1, or growth phase 1. And this is the functional part of the cell. This is the time where it's carrying out whatever its function is. So if it's a liver cell, it's going to be filtering out the blood. If it's a skin cell, it's going to be, protecting, creating a barrier in your skin, so on and so forth. Okay, so this is where it's doing the work. Okay, the second part is called S phase. S is for synthesis. And what's being synthesized is a new set of chromosomes through the process of replication. So you're doubling the chromosomes, well, you're doubling the amount of DNA in the synthesis phase. All right, and then the last one is called G2. And what's happening in this phase is preparation for mitosis. Okay, so this is where you're going to double the number of organelles. You're going to increase in volume and length. The chromosomes have already replicated, so that's already in place. And then towards the end, now you're going to prepare for splitting into two new cells that are exact copies of each other in mitosis. Yes?